Medical nanotechnology is a rapidly advancing field that holds great potential in revolutionizing healthcare and improving patient outcomes. In this comprehensive script, we will delve into various topics related to medical nanotechnology, exploring the latest advancements and breakthroughs in the field. From solid lipid nanoparticles to bio-inspired nanoparticles for drug delivery, we will examine the intricacies of each topic and discuss their applications in pharmacology and therapy. By the end of this script, you will gain a thorough understanding of the diverse applications of nanotechnology in medicine. So, let's begin our exploration of this fascinating field. Solid lipid nanoparticles, SLNs, have emerged as promising drug delivery systems due to their ability to encapsulate and deliver both hydrophilic and lipophilic drugs. These nanoparticles are composed of a solid lipid core surrounded by an emulsifier layer, providing stability and controlled drug release. The small particle size of SLNs enables enhanced drug absorption and tissue penetration, making them ideal for targeted therapy. Furthermore, SLNs have a high drug loading capacity and can be easily prepared using various techniques such as hot homogenization and cold homogenization. The versatility and biocompatibility of SLNs make them an attractive option for delivering drugs to specific sites in the body. Nanostructured drug delivery systems offer precise control over drug release and distribution, and self-assembly techniques play a crucial role in their design and fabrication. By using molecular self-assembly, these systems can spontaneously form ordered structures, such as micelles, liposomes, and nanofibers, which can encapsulate and protect drugs. The self-assembly process is governed by intermolecular forces, such as hydrogen bonding, hydrophobic interactions, and electrostatic interactions, allowing for tailored drug delivery systems that can release drugs in a controlled manner. Moreover, self-assembled nanostructures can be functionalized with targeting ligands to enhance their specificity and improve therapeutic efficacy. Nanogels are three-dimensional hydrogel networks with nanoscale dimensions that can encapsulate and deliver therapeutic agents. These versatile drug delivery systems possess a high water content, allowing for efficient drug loading and release. The controlled release of drugs from nanogels is achieved through stimuli-responsive mechanisms, such as changes in pH, temperature, or enzyme concentration. This enables site-specific drug delivery and minimizes systemic side effects. Nanogels can be synthesized using various methods, including emulsion polymerization and template polymerization, and their unique properties make them attractive for a wide range of biomedical applications. Quantum dots, QDs, are semiconductor nanocrystals that exhibit unique optical properties, such as size-dependent fluorescence and long-term photostability. These properties make QDs ideal for biomedical imaging applications, including single-molecule tracking, bioimaging, and fluorescence-guided surgery. In addition to imaging, QDs can also be functionalized with drug molecules, enabling targeted therapy. Moreover, the tunable properties of QDs allow for precise control over drug release kinetics and dosage. However, the potential toxicity of QDs remains a concern, and further research is required to ensure their safe use in clinical settings. Magnetic nanoparticles have gained significant interest in targeted drug delivery due to their ability to be remotely guided to specific locations in the body using external magnetic fields. By functionalizing magnetic nanoparticles with drugs and targeting ligands, site-specific drug delivery can be achieved, reducing systemic side effects. These nanoparticles exhibit superparamagnetic behavior, enabling them to respond to magnetic fields without residual magnetization. Various techniques, including co-precipitation and thermal decomposition, can be used to synthesize magnetic nanoparticles with controlled size and morphology. The combination of magnetic guidance and controlled drug release holds great potential in improving the efficacy and safety of drug delivery systems. Nanostructured hydrogels are cross-linked polymeric networks that swell in water and can encapsulate and deliver drugs. These hydrogels possess a three-dimensional porous structure, allowing for efficient drug loading, sustained release, and protection of encapsulated drugs from degradation. Their high water content and soft nature mimic the extracellular matrix, making them biocompatible and suitable for tissue engineering and regenerative medicine applications. Nanostructured hydrogels can be engineered to respond to external stimuli, such as temperature, pH, and light, enabling on-demand drug release. The tunable properties of these hydrogels make them promising platforms for personalized medicine. Gold nanoparticles, OMPs, have gained significant attention in theranostics, which combines diagnostics and therapy, due to their unique plasmonic properties and biocompatibility. OMPs can be functionalized with targeting ligands and imaging agents, allowing for simultaneous imaging and targeted therapy. The surface plasmon resonance of OMPs can be tuned to specific wavelengths, enabling them to act as contrast agents in various imaging modalities, such as computed tomography, CT, magnetic resonance imaging, MRI, and photoacoustic imaging, PAI. Additionally, OMPs can deliver therapeutic agents, such as drugs and CERNA, achieving localized therapy while reducing systemic toxicity. The versatility of OMPs makes them promising tools for precision medicine. Combination therapy, which involves the simultaneous delivery of multiple drugs, has shown great promise in treating complex diseases. Multifunctional nanocarriers, such as liposomes and polymeric nanoparticles, provide an ideal platform for combination therapy due to their ability to encapsulate different drugs with distinct physicochemical properties. These nanocarriers can be engineered to release drugs sequentially or in a synergistic manner, enhancing therapeutic efficacy. Furthermore, multifunctional nanocarriers can be functionalized with targeting ligands and imaging agents, enabling site-specific drug delivery and real-time monitoring of treatment response. The rational design of multifunctional nanocarriers holds immense potential for personalized medicine and improved patient outcomes. One of the key challenges in nanomedicine is achieving efficient penetration of therapeutics through biological barriers, such as the blood-brain barrier, BBB, and tumor microenvironment. Novel approaches, such as surface modification of nanoparticles with cell-penetrating peptides and use of external stimuli, have been explored to enhance the penetration and accumulation of nanomedicine at target sites. Strategies like ultrasound-mediated drug delivery and magnetic targeting can improve the site specificity and therapeutic efficacy of nanomedicine. Additionally, nanotechnological advancements, such as nanoparticle-coated microneedles and microbubbles, offer innovative ways to overcome biological barriers and deliver therapeutics to previously inaccessible areas. The development of robust techniques to enhance the penetration of nanomedicine holds great promise in expanding the scope of targeted therapy. Nature-inspired nanotechnology draws inspiration from biological structures and processes to design efficient drug delivery systems. Biomimetic nanoparticles, such as liposomes and exosomes, mimic the natural behavior of cells and play important roles in drug delivery and cellular interactions. Liposomes, composed of phospholipids, can encapsulate hydrophobic and hydrophilic drugs and mimic cell membranes, allowing for effective drug delivery and targeting. Exosomes, on the other hand, are naturally occurring extracellular vesicles that can be harnessed as drug carriers due to their ability to transfer biomolecules between cells. By leveraging these bio-inspired nanoparticles, drug delivery systems can achieve enhanced biocompatibility and efficacy, leading to improved therapeutic outcomes. Smart nanodevices are designed to respond to specific stimuli in their environment, triggering drug release at desired locations and time points. By integrating stimuli-responsive materials, such as polymers, hydrogels, and magnetic nanoparticles, into nanodevices, precise control over drug release kinetics can be achieved. External stimuli such as light, temperature, pH, and magnetic fields can be utilized to trigger drug release from these nanodevices. In addition to stimuli-responsive materials, nanodevices can be engineered with sensors and feedback control systems to tailor drug release based on real-time monitoring of physiological parameters. The development of smart nanodevices enables personalized medicine, ensuring optimal therapeutic outcomes and minimal side effects. Understanding the cellular and molecular interactions of nanomedicines is crucial for optimizing their therapeutic efficacy. Upon administration, nanomedicines interact with various biomolecules, such as proteins and cell membranes, which can influence their pharmacokinetics, biodistribution, and therapeutic response. The physicochemical properties of nanomedicines, including particle size, surface charge, and surface functionalization, play a key role in mediating these interactions. Techniques such as dynamic light scattering, electron microscopy, and fluorescence microscopy can be employed to study the internalization, intracellular trafficking, and fate of nanomedicines within cells. By unraveling the intricate interactions between nanomedicines and biological systems, we can refine their design and enhance their performance in clinical applications. In conclusion, medical nanotechnology offers immense potential for revolutionizing drug delivery and improving patient outcomes. The topics covered in this script, ranging from solid lipid nanoparticles to cellular and molecular interactions of nanomedicines, provide a comprehensive overview of the current advancements and challenges in the field. As researchers continue to push the boundaries of medical nanotechnology, it is vital to strike a balance between complexity, terminology, and detail to ensure effective knowledge dissemination and application. By harnessing the power of nanotechnology, we are poised to create targeted and personalized therapies that maximize therapeutic efficacy while minimizing side effects, ushering in a new era of precision medicine.